Today's topic is about the sterilization of carrier materials. In the first slide, we discuss about the sterilization process, particularly autoclaving. The autoclaving is an easiest way to remove the pathogenic microorganisms. And the pathogenic microorganisms are easily spoiled or contaminate the carrier material after packaging. That's why we must sterilize the carrier material. So the autoclaving temperature used for the process is about 121 degrees Celsius for 2 hours. This is the temperature almost we maintain in a pressure cooker. That's why we can use a pressure cooker as an alternate one for autoclave instrument. The sterilization of carrier material is essential to keep high number of inoculants bacteria on a carrier for a long storage time. If you want to store the carrier material for a long period, you must sterilize the carrier. So, there are 3 steps followed in the sterilization of carrier material. First step is to make a fine powder form or granular form of carrier material. For example, the rice husk ash must be sieved via different sieving filters. So, you may get a fine powder. And then neutralize the carrier pH by adding a calcium carbonate in the ratio of 1:10. For example, 10 gram of carrier is mixed with 1 ml of calcium carbonate. This is the standard point of making neutralization of the pH. Or it may be moistened by adding a water. The third step is about to sterilize the material by autoclaving or by gamma radiation or by sun drying method. In our lab, we prefer the steam sterilization process by using a pressure cooker. In an industry, you may go for autoclaving process or a gamma radiation. But there are some merits and demerits in autoclaving as well as the gamma radiation. So, these are all the essential steps in a carrier sterilization. In the previous slide, we discussed the sterilization methods. Now, we will see the step-by-step method of final product recovery. In a burning process, we can get 3 forms of composting materials. First, to get a black ash by taking a dried rice husk and placed it in an electrical furnace for 5 hours at 400 degrees Celsius. After that, we get a black color ash. For complete combustion, the ash is ground into powder and heated again at 600 degrees Celsius for 5 hours. Next, the pure amorphous white silica is also obtained by burning this rice husk at 800 degrees Celsius for 5 hours. Almost in 400 degrees Celsius we get black color ash, at 600 degrees Celsius get a complete ash, and 800 degrees Celsius you may get pure white silica. Care should be taken to prevent the loss of pressure after autoclaving. Here the sterilization is the best method and also conducted on 2 consecutive days. After sterilization, the carrier is packed in a partially open thin wall polypropylene bags and autoclaved for 60 minutes at 121 degrees Celsius. Here you should note that some of the materials in plastics produce toxic substances during autoclaving process, and sometimes the carrier medium is less effective because of microbial contamination. That's why the sterilization is very efficient one as well as a best method to prevent contamination. But the rice husk consume less energy for their production and also maintain as a sustainable resources for long period. That's why we prefer the rice husk ash for using biofertilizers. Next slide is about the types of sterilization method. Here we are using two types of sterilization method. One is steam sterilization, another one is ionizing radiation technique. In ionizing radiation, you have to use gamma rays only. That's why it's called a gamma radiation step. The first one, steam sterilization, it is an ordinary method. Here, the physical as well as the chemical changes may occur due to the plastic papers. But in ionizing radiation, there is no significant changes occurs during the radiation because it is an industrial method. That's why you may use a bulk amount of carrier material for sterilizing purpose. That's why we are using a gamma rays for the sterilization. Next, the steam sterilization is costly as well as time consuming and easy to handle. The gamma radiation technique is cheaper as well as time saving and efficient handlers are needed for this technique. And the steam sterilization is easily available in the market because there is no rules and regulation for the steam sterilization technique. But in gamma radiation, there is a limited availability in the market, and also you must follow the rules as well as the regulation in a strict format. That's why this is a limited available resources in the market. Here we discuss about the parameter changes after sterilization. The parameters like post-operative chance of contamination, second distortion of packaging material, third extent of throughputs, and fourth one mode of action. Fifth one is about the output of the result. The first one, post-operative chances of contamination. After sterilization, there are so many risk may arise due to the moisture as well as the microbial load. In a steam sterilization, the packets must be sealed before sterilization process because after sterilization some of the moisture may enter into the packets. That's why we must seal the packets before sterilization. In ionizing radiation, there is no chance for the contamination because the packets are sealed as well as irradiated. So there is no repackaging for this method. And the second one, distortion of packaging materials. Here the distortion may caused due to the steam sterilization. Because of the plastics or heat labile molecule, that's why distortion may caused by steam sterilization. But ionizing radiation, there is no risk. And the third point is extension of throughput. It's comparatively less as well as only a small volume may be sterilized at a time. But in ionizing radiation, comparatively it's a higher efficient and large volume can be sterilized. In a mode of action, the proteins of the microbial cells are inactive or died under moist heat condition due to the process of coagulation of protein. But in ionizing radiation, the DNA of the cells reacts with the radioactivity compound and then turn into inactive stage. This is called cold sterilization or dry heat sterilization process. The last one is about the output. Just maintain less number of cell over long storage period by steam sterilization method. But in ionizing radiation, you have to maintain higher number of cells over a long storage period. These are all the advantages as well as some of the disadvantages in steam sterilization as well as in the ionizing radiation. This slide is about quality specification. There are so many characteristic specifications followed before and after sterilization. In before sterilization, you should check the form of base. This is nothing, it's called carrier. It may be in the form of powder or liquid base one. Suppose if it is a powder form, example, rice husk ash, it must be moistened with water. After sterilization, we should follow the remaining tests. First, check the contamination level by plating technique for carrier material. In the powder or the granular form of carrier, the colony forming unit must be 5 into 10 power 7 cells per gram, and in the liquid form it must be 1 into 10 power 8 cells per ml, which indicates the minimum range of contaminants. Next, the viable microbes or live inoculants is detected by serial dilution technique. This technique is suitable for detecting the number of contaminants. Particularly in the dilution of 10 power 5, if you are not observing any contaminants, then go for pH range, particle size and moisture content detection. Here the final range of pH must be 6.5 to 7.5. Particle size is about 0.15 to 0.212 mm, and the moisture content is about 30 to 40 percentage. Finally, this is ready for mixing with carrier.